14.3. So looking at how do we process messenger RNA? Now, important comparison between prokaryotic and eukaryotic cells. Only in eukaryotic cells do we process the RNA. To remind you of where we came from, like bigger picture what we're doing, we talked about the central dogma. The central dogma says, how do we go from DNA to making proteins? How do we use the genes that are organized in DNA to make RNA? We make a copy of DNA called RNA, and then we take that RNA and make proteins. The process of taking DNA and making RNA, that's transcription. And the process of going from RNA to proteins is translation. So what 14.3 is talking about is, what is the step between going from RNA to proteins? In eukaryotic cells, we need to process that RNA. We need to make some adjustments to that RNA so that the process of translation can happen smoothly. Okay, and I talked about this when I did the kind of the overview of protein synthesis, where basically we remove certain portions of the RNA. So let me show you a picture of it to help you understand what's going on. So here is our RNA molecule that we made. Now, if we look right here, we have the protein coding segment. Really, like what we need the RNA to do is to make proteins. But what you see here is only actually a fairly small portion of that RNA technically codes for that protein. There's a lot of other things in that RNA that are used to, basically used for like leading to the assembly of the ribosome to make that protein. So first thing I want to talk about is, remember the start and stop codons? This is what's going to be used to tell the ribosome, where to start. So the stop codon, start tells it where to stop, right? Then you have something called UTR. UTR, these are untranslated. Why is there two T's? Untranslated region. This is where ribosome binds. The ribosome binds there. And then we have a 5' cap. Okay? 5' cap. And it's made of a guanine nucleotide. I don't know if the AP exam would really care too much about that, but go ahead and make that something you remember, especially if you study this in college. Somebody would probably care about it. That the cap is made of a guanine nucleotide. 5' cap. And a lot of these modifications, involve, like including this, it helps with protection. So we want to protect this mRNA from hydrolytic enzymes. What's a hydrolytic enzyme? You remember? So all around the environment that this messenger RNA is existing, there are hydrolytic enzymes. Why is that like, whoa, Luke?
Basically, break up the
Yeah, so when we talked about monomers and polymers, I said if you want to break a polymer down to monomers, you could use hydrolytic enzymes that break things up. Well, we don't want to break this mRNA up, so this cap and even this tail, bless you, this poly A tail here, these both will function helping in protection. This is also protection from those hydrolytic enzymes. Okay. Talking about the poly A tail, they call it poly A because you have all these repetitions of the adenine nucleotide A's. Okay. There's also a 3' UTR, untranslated region. So those are used for protection. They'll be used for to help assemble the ribosome and other proteins that help in translation. And then the third thing is facilitating export to the cytoplasm. So these modifications like the 5' cap, the poly A tail, basically other proteins can bind. So the third thing would be proteins bind for transport. To transport this from the nucleus to the cytoplasm. Okay? Because we make this messenger RNA in the nucleus, but the ribosomes are in the cytoplasm where we actually will use those ribosomes to make the proteins. Okay? All right, let me make sure I said everything here. You get 5' cap, and it's a guanine nucleotide. 3' you get the tail. Oh, here's how you can remember. 5 prime G. 5G. 3, like, imagine you're playing basketball and you hit a 3 and everybody says, hey! I don't know. I'm open to suggestions. Okay.
Sometimes, like, I remember these things just because of, like, the awkward moment of, like, after the joke.
I don't know. I have a very horrible memory. The only way I can memorize, there's a lot of things you gotta memorize about. The only way I can memorize them is just I gotta think of some dumb way. Because some things you can just understand. Other things like I don't know, you just gotta memorize it. So, okay, moving on. So here's our messenger RNA. Now this is pre-mRNA. Pre means it's before we've processed. So now we gotta process it, and what we do is we cut out the introns. Now, you would think, at least in my mind, you would think that the introns would stay in and the exons would be the ones taken out. But that is not how they named them. Here's what they were thinking. They were like, introns, we call them introns because they are interspersed, if that's how you spell that, regions of mRNA that are not expressed. Exon is for expressed, as in gene expression. Gene expression is saying genes are on DNA. Gene expression is when you use that gene to make proteins. So only the exons, the things that stay in, so here's the exon, only the exon will be expressed. The intron are the interspersed regions of DNA that are not expressed. That's where the naming comes from. But again, it's kind of backwards, so I think what is more obvious, which is in versus exiting. So notice your 5' cap, 3' poly A tail. Now, another thing with these is there is alternate splicing. Basically, we can change how we imagine like this as like your book compares it to like a movie. I don't know if you ever edited a movie on your laptop or your phone. It's like you're editing out, like the introns are like editing out like the transitions, the parts of a film you don't need. Well, you can imagine you can make a completely different film by like not editing out some of those things. So alternate splicing, you can make different proteins by changing what is an intron versus what is an exon. That's a really important idea. And that helps with efficiency. So that raises efficiency because one RNA transcript can make many different proteins. So one mRNA can code for more than one. I don't know why my end's all goofy like that. One mRNA can code for multiple proteins. So we change which ones are exons versus which ones are introns. That will change what will ultimately become the protein. Thomas? Yeah. Oh, untranslated region. So it's a region that's not going to be translated. Basically, this is used to kind of assemble and deassemble the ribosome. So I'll kind of show what those, in 14.4, I'll kind of show where those come in. Okay. All right, let me make sure I said everything. So again, that whole process called RNA splicing. Anybody ever see that movie Spliced? It's like that creepy movie. It's like this guy, he could change his DNA to make himself become like a monster. It's like I think it's part of like the Marvel universe.
Wait, is it animated?
It's not animated, no. It's actually like really creepy. It's like Patricia, and maybe it's not called Spliced. I don't know. Could be some different. Anyways, whatever. Yeah. Anyways, how we do splicing is you use something called a spliceosome, which is conveniently named. Now, a spliceosome, it's made up of mostly RNA plus some protein. Why I emphasize the RNA portion of this is because most enzymes are what? Made of what? What are most enzymes? What biomolecule are most enzymes made of? Luke? Proteins, yeah. Most enzymes are proteins. But a spliceosome is kind of a rare case where you can have a molecule where most of the enzyme, the enzymes speed up chemical reactions, most of the enzyme portion of the molecule is actually an RNA. And here's why it's actually kind of beneficial that it's made of RNA. Because here they're showing like an interior portion of the spliceosome. This spliceosome can complementarily base pair with the corresponding DNA. So let's say on this portion of the spliceosome it's ACU. What would it be on this portion of the intron that it's going to splice out? Think of the base pairing rules. What does A go with in RNA? Apple in the tree, but in RNA it's U and G and A. So the RNA portion of the spliceosome would recognize the UGA of the intron, and then it would be able to then essentially do that all along the intron and use that to know what part of the mRNA to cut out, what parts to leave in. And again, if I wanted to make a different kind of protein from the same mRNA, we switch which ones are exons, which ones are introns. Pre-mRNAs before the spliceosome done its job. mRNAs when you actually have the spliced components. And again, this only happens in eukaryotic cells. Prokaryotic cells, they don't have spliceosomes. They just they do the RNA and they send it right to the proteins. Okay. All right, let me make sure I said everything. So alternate RNA splicing, again, that's where we can change which things are introns, exons to make more than one protein. Yeah, ribozymes. So the spliceosome. There's no way that's right. Did I get that? That just looks goofy to me. Spliceosome is an example of a ribozyme. A ribozyme is an enzyme made of ribosomal RNA. Okay? So again, it's just this idea that they didn't know before until they discovered the spliceosome. They didn't really know about like the idea that like you could have an enzyme made of RNA. And that kind of gets into significance with like sort of like origin of life research and things that we might be able to touch on a little bit for the AP exam, but it's kind of a sort of a revolutionary idea. And another thing that our cells don't do this, but some organisms like protozoa, their introns can actually splice themselves. So if I were to like, how that possibly could work is the introns of protozoa, let's say this is like A, G, C right here, it could then recognize UCG, or better yet, this would be GCU at the other end of the intron, and so then those can complementary base pair and cut themselves out of the mRNA transcript. It's kind of a wild idea. Anyways, just be aware that some organisms can do that, can do self-splicing, they call it. So self-splicing. That's when the introns can catalyze their own splice. They can cut themselves out. Again, our cells can't do that, but some can. All right, we good so far? Okay, yeah. So as I said, pretty short for first section.